Hi everyone, and welcome to our lesson today, which will focus on lipids. Now, the first thing you have to ask yourself, well, what exactly is lipid? Okay, now lipids, you usually hear them called fats or various forms of fats. And ultimately, the definition of a lipid is based on its solubility because lipids are very almost nearly not soluble in water, but they're readily soluble in organic solvents such as chloroform or acetone. Okay, so when you think of lipids, when you've seen like oil doesn't mix with water, that's because they're not readily soluble in water, but instead are soluble in organic solvents. Now, there are two groups of lipids I'd like you to know. They're based on their chemical structure. So lipids can either be considered, let me get the laser point going, open chain lipids, which are the ones that you see on this side, which includes the saturated and unsaturated fatty acids, things like triacylglycerols, sphingolipids, phosphoacylglycerols, and glycolipids, all of which we're going to discuss in today's lecture. Or the chemical nature of a lipid can be the fused ring structure. So you see fused rings instead of long chains. And the fused rings are the steroid examples that we're going to go through in this lesson. They include cholesterol and various sex hormones and other hormones that we're going to mention a little bit later. Now, as I promised, we're going to go through each of the different groups or categories of lipids that we just mentioned. The first one is the fatty acids, and there are a few different things you should think of when you think of fatty acids. First of all, you should think of their structure, what two components they're made of. And don't worry about, trying to jot down as I'm speaking. I'm going to give you a recap on the next slide of exactly what I want to write, what I want you to write, so you can just listen and look right now. So the two components that you will find with fatty acids are their polar end, which when you see them drawn out like this, it looks like a round little head. And then you see the long chain. So they have a polar head that's a carboxyl group. And then they have the long hydrocarbon chain. Okay, so over here you see the carboxyl group, which is COOH, and you have the little code down here, the color coding. And then this is the long chain of hydrocarbons. Okay, so that's the two components. Now you'll notice that I said the polar head and the nonpolar hydrochain tail. That is why we call fatty acids amphipathic. Amphipathic is when a molecule has both hydrophilic end and a hydrophobic end. Okay, so in the case of the fatty acids, the carboxyl group is the hydrophilic end, and then the hydrocarbon tail is the hydrophobic end. Okay, so you can think hydrocarbon tail is the hydrophobic. Now, you'll notice that before on the previous slide and on this one, you see the term saturated and unsaturated fatty acids, and I'm sure in your everyday life you've always heard about these terms as well. So when we talk about the differences between unsaturated and saturated fats, there are a few different things that I want you to be aware of. First of all, you'll notice that the unsaturated fats in this figure, they contain at least one double bond. Okay, so if it has a double bond, it's unsaturated. That double bond can be broken open and then that can become saturated. But while there's a double bond, that is what makes it unsaturated. Whereas the saturated fatty acids, it's saturated because there is a hydrogen at every possible bond. There's no double bond present for you to break. So that's the big main difference. A few other differences that I want you to be aware of is because of the double bond in the unsaturated fatty acids, you can have either trans unsaturated fatty acids or cis. And so you notice that the trans configuration here versus the cis same side configuration here around the double bond. And so we'll talk about later, what it means in terms of trans fats and saturated fats, unsaturated, the healthy and the unhealthy information. But for now, be aware of that difference. The other thing I want to mention in terms of differences is the unsaturated fatty acids. They tend to be liquid at room temperature, and they have a lower melting point, whereas the saturated fatty acids are more the solid fats that you'll find at room temperature, and they'll have a higher melting point. And part of the reason why saturated and the unsaturated trans fatty acids are more problematic to your health is that when you look at the structures of them, they're more linear, okay? The cis bond or the cis conformation of this third fatty acid gives it a kind of curved, bent shape, whereas these two are linear, which means that they can stack more easily and accumulate more heavily, which can create plaques that we're going to talk about in later slides of this lesson that are very dangerous for your arteries and your heart health. Now, as I promised, here's one of those recap slides, basically giving you the notes that I would like you to have for that previous slide that we just went over. Again, there'll be a recap slide in a minute, so you don't have to worry about if you don't catch the notes right away. The next category of lipids that we're going to talk about are the triacylglycerols. Now, when you see that it has glycerol in its name, so glycerol here, what glycerol is this chemical structure right here. You'll see that it's a compound that has 3 hydroxyl groups. Then what happens is if ester linkages occur, where each of these 3 hydroxyl groups end up linked to a long chain of fatty acids, that's when you end up with a triacylglycerol. Okay, and these are very, very important because a lot of the lipids that we'll talk about are parts of membranes, but these guys are not part of membranes. Instead, their role is as concentrated storage of metabolic energy. And later in this lesson, we're going to go through how you then can access this energy. But for now, focus on when you hear triacylglycerols, sometimes also called triglycerides. Their main function is storing metabolic energy, a means of storing fatty acids, basically. And later on, when we talk about actually using these triacylglycerols, the enzyme that we'll talk about that hydrolyzes them is lipase. Now, another thing that I want to mention when we talk about triacylglycerols is that sometimes you'll hear the term saponification, and that's basically because with the reactions that can occur with triacylglycerols, you're able to make salt products that are soaps. So saponification is when you use fats to make soaps. And if you ever saw the movie Fight Club, then you will have seen all about this. Okay. Now, as promised, this is the recap of the notes from the previous slide. The next lipid that we're going to take a look at and talk about is the phosphoacylglycerols. Now, the phosphoacylglycerols are similar to what we discussed in the previous slide. So it starts with a glycerol. So this is to remind you of that structure. But the difference now is instead of 3 fatty acid chains, this glycerol gets an alcohol group replaced with a phosphoric acid molecule rather than the carboxylic fatty acid. So instead of having the 3 alcohol groups replaced by 3 fatty acids, in the phosphoacylglycerol, you get one, so you can see it here, one of the phosphoric acid molecules attached, and then the other two are fatty acid chains. You can see it here and here. And specifically, you'll notice that one will be a sterile group and the other a linoyl group, such as from, linoleic acid. But you don't have to memorize that. I just want you to see the difference in terms of, instead of 3 fatty acids, now you're going to see the phosphoric acid group and 2 fatty acid chains. And please know that whereas the previous slides we mentioned a significant function in terms of energy storage, now the phosphoacylglycerols, their significant function is being part of biologic membranes. So we're going to see this when we talk about the lipid bilayers later on. The next lipids that we're going to take a look at are the waxes and the sphingolipids. First up, we have the waxes, which I focus on in the figures on this side of this slide. Waxes are basically composed of complex mixtures of long-chain carboxylic acids, which are, the fatty acid structures that you'll see hanging off, and the long-chain alcohols. So as you can see in this example, you have the fatty acid here and then the long-chain oleic alcohol. And they're very important in terms of being protective coatings. You find them for both plants and animals. And so you've all seen something waxy and you know how hard it might be to penetrate that kind of a coating. So it does serve that function well. Now, the other one to mention on this side are the sphingolipids. And unlike some of the other lipids that we were just talking about, there's no glycerol in the sphingolipids. Instead, now you're going to see with the fatty acid chain, you now have what's called sphingosine. And sphingosine is simply a long chain amino alcohol. Okay, and that's what makes this guy a sphingolipid. And whenever you hear the term sphingolipids, I want you to think of the nervous system. Sphingolipids are very abundant in the cell membranes of the brain and various nervous system tissues, and they're found heavily in the myelin sheath, which you can see around each of your nerve cells. Okay, so when you hear sphingolipids, please associate that one in terms of the nervous system, especially the myelin sheaths around your nerve cells. The last of the lipid category examples that I want to go over are the steroids. And steroids is a term that most students have usually encountered. As you can see by the figure, any of the steroid lipids are defined by having that fused ring structure instead of the long carboxylic acid chains or hydrocarbon chains that you saw in all of the previous examples. And you'll notice that they all have the same general structure in terms of their fused rings look very similar to each other. They have the same pattern of having 3 of the 6-membered rings and 1 of the 5-membered. So 3 6-membered rings attached to 1 5-membered ring. And there are very important examples, including, as you see in this figure, cholesterol and these sex hormones. And there are also other examples, things like the mineralocorticoids and steroids like aldosterone and cortisol, that you'll discuss in a lot more detail in later lectures and in other courses as well. So even though I focus here because of the figure on cholesterol and sex hormones, please be aware those are not the only steroids, and ones like aldosterone and cortisol will come up in a lot of discussions as well in other classes. Now that we've talked about the different types of lipids, it's important to go through their functionality and their significance in structures such as the biological membranes. Now, as we mentioned a few slides ago, the phosphoglycerides, out of all of the groups of the different lipids we discussed, those are the ones that are the principal components of the membranes, including our own plasma membranes around each of our cells. Now, the reason why these are such important lipids, and especially for membranes, is the amphipathic nature that we talked about earlier. So the fact that they have a hydrophilic end and a hydrophobic end allows them to arrange as these lipid bilayers. So lipid bilayer meaning you have a lipid layer here, and opposing it, you have the second layer of lipids here. And because they're amphipathic, it lets you have the hydrophilic polar head groups in contact with water, and then the hydrophobic tails hidden away so that they're not exposed to water. And you can see that in this structure here too, the way the bilayer allows for the inner compartment and the outer surface to be in touch with water, and then the inner region of the membrane with the hydrophobic tails gets nice and hidden away. Please also note that the difference in terms of something that's just a lipid bilayer versus our membranes is that a lipid bilayer is strictly lipids. Okay, so what you're seeing in this figure here, you only see lipids here. Whereas our plasma membranes instead, when you see the word membrane, it not only has the lipids, but it also has the embedded proteins in it. So just be aware of that terminology, that a lipid bilayer lacks proteins, whereas a membrane is the lipid bilayer plus the embedded proteins, which can be 20 to 80% of the membrane weight. And when we think of these lipid bilayers and these membranes, there are various aspects that can influence how rigid or how fluid these structures are. Now, when it comes to this rigidity or this fluidity, those being opposite terms of each other, ultimately the arrangement of the lipids and the exact composition of the lipids in this layer greatly affects this rigidity and the properties of this membrane. So for instance, I want you to know the types of lipids that will increase rigidity, which are cholesterols and saturated fatty acids, whereas unsaturated fatty acids, they will decrease the rigidity of the membrane, meaning that they will make it more fluid. And basically the reason why that is if you picture saturated fatty acids, for instance, we saw that they're very linear, right? They don't have any of the double bonds, they don't have any kinks in them. So when they're linear, they'll be very tightly stacked next to each other, kind of like you can see in this figure. More linear fatty acids get to be tight and piled together, whereas unsaturated fatty acids, especially the arrangements that you kind of can see here, if you picture the kink that we saw in terms of the unsaturated fatty acids, well, now you have a loosening of the membrane structure. You have the lipids more spread out from each other, which makes it more fluid rather than rigid. Okay, and I want to point out, so in this figure here, this is where you can see how cholesterol can influence the structure as well. Now, when we talk about composition, I want to point out that in addition to composition affecting the rigidity of these structures, also temperature can affect the fluidity of a membrane, of a lipid bilayer, because with heat, the very ordered structured bilayers become less organized. And the bilayers that are basically already comparatively disordered, they become even more disordered. And that's actually what this figure down here is meant to show you. As you increase heat, membranes can go from a gel kind of phase to what's called a liquid crystal phase, which, as you can see, is a more fluid, spread out phase rather than the very rigid, tight gel formation here. Now, even though the lecture is called lipids, and I said we would focus on lipids, it's also about the membranes that those lipids are such big players in. And as I mentioned before, when it comes to our membranes, what makes it a membrane rather than just a lipid bilayer is the fact that it has proteins within it. And there are two categories of membrane proteins that I want you to know the difference between. There are peripheral proteins, and there are integral proteins. So peripheral, something that's on the periphery or peripheral, is just on the surface. So in this figure here, heterotrimeric protein G is a peripheral protein because you see here, heterotrimeric, it has the three subunits of the red, the yellow, and the blue in this picture. And all of those subunits, this whole protein, is just on the surface of this membrane. Whereas rhodopsin is the example of an integral protein because integral proteins are embedded within the lipid bilayer. So you notice that the purple protein in this figure spans across the entire lipid bilayer because it is embedded in, it is an integral protein. And the difference in terms of their locations also greatly affects when we're dealing with that membrane, because as you can see, if something is just a peripheral protein, it's going to be pretty easy for you to remove it from that membrane, usually with mild treatment, something like changing the ionic strength of the medium, for instance. Whereas if something is integral, it's really embedded within that membrane. So you can't just get rid of it from the membrane without very difficult, harsh conditions like adding something like a detergent or using something extensive like sonication. And you end up basically destroying or denaturing that protein. And even then, you still have difficulty removing it from that membrane. Whereas the peripheral one is a lot easier for you to get rid of from the membrane, and it's usually just attached by something like, electrostatic interactions. Now, when we teach about proteins, I usually in my other classes refer to them as basically rock stars. They're the type of biomolecule that has a ton of different functions. And even in their role as membrane proteins, they're not just doing one thing. Membrane proteins can have a lot of different functions, and I love this figure because it really captures a lot of the most significant functions. Some of the ones that your textbook will talk about are the transporter roles in terms of getting ions and molecules across the lipid bilayer part of the membrane. There's also cell surface receptor roles, which are very important in terms of cell signaling and really kind of understanding extracellular signals like those carried by things like hormones or neurotransmitters. And additionally, their enzymatic roles are very significant in our course because, as we saw in the first of the lectures that I did with you, enzymes really control everything within cells, all of the reactions. So it's significant that membrane proteins can have enzyme activity as well. There's also some of the other functions that may not be mentioned as much in this chapter, but are mentioned in significant detail in a lot of the other classes. The fact that membrane proteins can be surface identity markers and involved in adhesion and attachment. And during the COVID pandemic, cell surface identity marker membrane proteins are significant because things like that spike protein that you hear with regard to COVID vaccines and the antigens that can get recognized, the antigens of a virus or a bacteria that can get recognized by your antibodies and your immune system. those are cell surface protein markers. Okay, so next to this one on the figure, you can write that we mean antigens, A-N-T-I-G-E-N-S, antigens. Okay, so there's a lot of different functions. Now, when we talk about membranes and membrane structures, one of the terms you'll constantly hear is the fluid mosaic model to describe our plasma membranes. And the reason why it's called a fluid mosaic model is that the term fluid tells you that the membrane is not a rigid, solid structure that's unchanging. Instead, the term fluid implies that there's some motion to it, okay? And basically the proteins within it or on top of it can kind of float and move along the plane of that membrane. And you've seen in probably videos and images throughout your biological career of the membrane not being rigid, of it kind of moving or swaying and having you know, different abilities to change itself. Then the term mosaic basically means when you have different components together side by side that are not, you know, creating or merging into a new substance. And so the reason why this is a fluid mosaic model is not only is this structure, you know, fluid and not rigid, but you have a mosaic of all of those different embedded proteins throughout it. Okay, so the proteins that we discussed previously being embedded within it and on the surface, that makes it a mosaic because you have the proteins and the lipids side by side together. Now that you're comfortable with the components or the structure of the membranes and those proteins within them, it's important to think about the actual function. And one of the main functions when it comes to your plasma membrane is to be able to transport things across into and out of the cell. Now, there are different forms of transport that I want you to be familiar with. There is passive transport and there is active transport. And when you think about passive or active, think about energy, right? You know, does the process require energy from the cell or not? And so if you are an active person, you know, you're doing a lot of things that requires energy. So it makes sense that active transport requires energy. Now, the reason why a cell would either need or not need energy is how they're moving molecules or the solutes or the particles that are going across the membrane. So passive, you're moving molecules or solutes from high concentration to low. Okay, so as you can see in this side of the figure, you're moving these particles into the cell. They're in higher concentration outside compared to inside. So you're going with the natural gradient, right? Because things naturally want to move from high to low until they're balanced out at equilibrium. So this doesn't require energy because it's how these particles want to travel, right? The only difference in terms of passive transport forms is that it can either be simple diffusion, which is the one on the left, or facilitated diffusion. And you'll notice in the picture, the only difference is whether or not there's a protein channel that's helping, or protein carrier molecule or structure that's helping carry these particles, these solutes, these molecules across the membrane. Okay, so as you can see in facilitated diffusion, you need the extra help. It's helped along. And so notice, even though you're using this carrier protein to help the movement is still high to low, so it doesn't actually require any energy. And one of the examples that they use in textbooks a lot for this facilitated diffusion is glucose passing into an erythrocyte. That's an example of facilitated diffusion, whereas small uncharged molecules like O2, N2, CO2, any of those passing through the membrane, they're going to go through simple diffusion. Now on the other side of this figure, you have active transport. Now you notice ATP is present, right? Because this one requires energy. And the reason that it requires energy is because now you're going against the natural gradient. Okay, it's when you are moving particles, molecules, any kind of substance from a low concentration to a high concentration. Okay, so now energy is required. And an example of that you'll see in later lectures is the sodium potassium ion pump. An important clinical example of receptor proteins, doing their job is this figure here of the LDL receptor. And we're going to talk about LDL and cholesterol later on in this lecture. But right now, this is just to show you that there's an LDL receptor that helps to bind LDL to take it into vesicles within the cell to then transport it outside and basically, get rid of that LDL and, return it to the outside of the cell. And what happens is if you have too much cholesterol, then that excess cholesterol will inhibit the synthesis of the LDL receptors. And so if now you're going to have fewer LDL receptors, that means that you'll get less LDL transport or collection into vesicles and transport out of the cells, and you'll end up with elevated levels of LDL and cholesterol. And what that does, as you're going to see later on, is increase your risk of heart attack. Okay, so receptors can be very important in terms of proper function of your cells and of your body. Now, even though we've been focusing on proteins in terms of their connection with lipids, it's also really important to mention some vitamins that are called lipid-soluble vitamins, because these guys are not going to be like the water-soluble vitamins, like vitamin C, where you can just take as much as you want and then the excess will get excreted in your urine. Instead, lipid-soluble vitamins will be absorbed in fats. And so they end up getting stored in your body. And so when it comes to the lipid-soluble vitamins, please be careful and remember these four, because if you end up taking in an excess of these, you can end up having toxicity symptoms and issues in your body. So the four lipid-soluble vitamins to remember are vitamins A, D, E, and K. A is involved in your vision. D is involved in calcium metabolism, and you'll see a lot of people end up with vitamin D deficiencies and don't even know it. But some people with vitamin D deficiencies can show things like insomnia and depression, mental health issues, and fatigue as well. Then you have vitamin E, which can have major antioxidant properties, very good for your skin. And then vitamin K, that's involved in blood clotting and can also help in terms of the absorption and use of some other vitamins like vitamin D. So for instance, you'll notice sometimes when you take a vitamin D supplement, it will be paired with a vitamin K to help better utilize the vitamin D. Another group of compounds that's important for us to discuss, especially for those of you going into the medical field, are prostaglandins and leukotrienes, and you may have even heard of some of these already. So first up we have prostaglandins, and keep in mind this is another one of those slides where I'll have a recap slide coming up next, so don't worry about trying to write everything down. So the first thing I want to talk about when we look at prostaglandins, they're derived from fatty acids, specifically arachidonic acid, and you're going to see that come up for leukotrienes as well. Now, prostaglandins have a whole bunch of different functions in your body. They're especially involved in things like control of your blood pressure, your smooth muscle contractions, and inflammation in your immune response. And so certain medications that target things like inflammation and pain will inhibit prostaglandins. So for instance, aspirin inhibits the synthesis of prostaglandins, and also cortisone or certain steroids have anti-inflammatory properties because they are inhibiting prostaglandin synthesis. Now, you also have leukotrienes that are derived from that arachidonic acid as well. They're found in leukocytes, which are white blood cells, hence why they have leuko in front of their name. And when it comes to leukotrienes, the thing I want you to associate with their function is constriction of smooth muscle, especially in your lungs and in your throat, your respiratory system, because that's why if you ever have a severe allergy, like anaphylactic shock or an asthma attack, and you're having difficulty breathing, it feels like your throat's closing off, you can't get air, that's in part because of leukotrienes. And that's why I have this picture here, because what can block leukotrienes is some asthma medications. So in the inhaler will block the synthesis of leukotriene. They also have other drugs as well that are in development or in use for the treatment of asthma that are not only to block the synthesis of leukotriene, but also to block the leukotriene receptors as well. There's also a third class of these derivatives of arachidonic acid that I did not put on this slide. It's called thromboxanes. You're going to see it in the recap slide. They're also involved in smooth muscle contraction, and they also induce platelet aggregation. So, part of clotting and clumping of the blood. Now, you'll notice smooth muscle contraction came up in all three terms. All of the derivatives of arachidonic acid had that aspect. The last thing that I want to point out on this slide are the dietary options that can really help you in terms of inhibiting these particular lipid derivatives, which is helpful to you because, like we said, these are involved in inflammation, okay, and smooth muscle contraction, and you do not want excess inflammation in your body. Sure, it's fantastic as a defense mechanism when you're fighting an infection, but it is not good to have a lot of inflammation in your body on a regular basis. So eating foods that are high in omegas, especially omega-3 fatty acids like you find in salmon, that helps inhibit the synthesis or the formation of prostaglandins and the thromboxanes that I mentioned, especially since they have similar structures to each other. And if you're eating foods that are high in omega-3 fatty acids that will block the prostaglandins and thromboxanes, that means that you're helping to block the functions that I just mentioned, things like excess blood clotting issues. You will be reducing your potential for artery damage and heart attack or strokes because you're reducing excess clotting, you're reducing inflammation and smooth muscle contraction. So having a diet high in omega-3 fatty acids is very beneficial, and you'll see that come up in later slides as well. As promised, here is the recap of the notes that I wanted you to jot down from the previous slide. And here is how you spell thromboxane A, which is the third of the arachidonic acid derivatives. So arachidonic acid derivatives are prostaglandins, leukotrienes, and thromboxane A. Okay, so now that you're all experts in terms of lipids and membranes, now we're going to start looking at metabolic reactions and metabolic activities. And we're going to spend the next portion of this lecture looking at metabolic oxidation of lipids. And that's very important because that allows the large release of energy through production of things like acetyl-CoA, NADH, FADH2, all of these things that I'm sure you've heard about in other classes when you're looking at things like electron transport chain and citric acid cycle and all of that in terms of ways of obtaining chemical energy or accessing stored chemical energy. So now, when we think of the metabolic oxidation of lipids, the first thing that I want you to know is the chief source of energy that's going to come from this breakdown of lipids is specifically the oxidation of fatty acids. And this makes sense because if you think of the fatty acids, there's a whole lot of chains of hydrocarbons. There's a whole lot of potential energy in those long, long chains that can be broken down. Now, the way that this is going to be done is through enzymes called lipases and phospholipases. Okay, and it makes sense again because when you hear those enzyme terms, they are basically named for what they are doing. And the reason why you see both here, including the phospholipases and what you see in this figure here, you see both lipases and phospholipases, is because when we are looking at the oxidation of energy, we mentioned earlier the big chemical energy storages that you can have in terms of lipids. And so triacylglycerols, which you see here, triacylglycerols, we've mentioned before, are the main storage form of the chemical energy of lipids. And the phosphoacylglycerols, which are important components of biological membranes, they're also going to have fatty acids as part of their structure that can be broken down if necessary. So now when we talk about fatty acid oxidation and metabolism, first the fatty acid has to be transported into the mitochondria. Now in order for this to happen, the first thing that has to occur is activation. And for activation, you basically have the formation of a thioester bond between a fatty acid and acetyl-CoA. Now, I don't want you memorizing all of the steps or anything that we're going to go through in metabolism and in the figures that you're going to see. I'll highlight in the bullet points here the main points that I want you to know, but I'll then talk a little bit to make those concepts clearer. So as I was saying, activation in lipid metabolism is the formation of a thioester bond between a fatty acid and acetyl-CoA. And now when you have acyl-CoA formed here, that can cross the outer mitochondrial membrane, but it cannot cross the inner membrane. So in order to have the fatty acid shuttled into the mitochondria, you then need carnitine. And there's a whole series of events in order to have that carnitine serve as a shuttle to bring the fatty acid into the mitochondria. Again, I'm not going to have you memorize any of that, but I want you to recognize and circle star highlight that term carnitine, as that is the shuttle that allows for the fatty acid to be carried or transported into the mitochondria so that you can then move on to the oxidation within the mitochondria. So once you've had the activation and the transport of the fatty acid into the mitochondria, now you can get to the beta oxidation of the fatty acids. And as you can see here, we have the full cycle mapped out for you. There are 4 main steps. So you see the 4 steps are oxidation over here, hydration, oxidation again, and cleavage. And you'll notice that we mentioned transport into the mitochondria because the mitochondria is the main organelle where you have this beta oxidation cycle taking place. But even though that's the main location, I do want to note for you that sometimes they've found some other sites of beta oxidation to a lesser extent, specifically in peroxisomes and glyoxysomes. But the main site of beta oxidation will be the mitochondria. And the clinical significance when you are looking at this cycle is that there's actually a category of drugs known as hypolipidemic drugs that I want you to know. And when you see the term hypolipidemic drugs, when you see hypo, that means less. So you want less lipids with the function of that drug. And the reason why it's less lipids is because it is a drug used to try and control obesity. And the way that it does that is by stimulating beta oxidation in peroxisomes. And so As you can see, beta-oxidation is the ultimate breakdown of lipids, of fatty acids, and metabolizing them. And so that will help with obesity, reducing your storage of fat within your body and your storage of lipids within your body. And I want you to note that you'll also see that throughout this cycle, you have the FADH2 and the NADH being produced as byproducts that I mentioned earlier, also acetyl-CoA. And seeing these as products within this oxidation is important because, as you all know, you've heard these terms with regard to making energy. So each of those can then be used in terms of obtaining ATP in other cycles. Now, speaking of other cycles that give you energy, mentioning the NADH, the FADH2, the acetyl-CoA, it's important to kind of compare the breakdown or metabolism of lipids versus the breakdown of glucose or sugar in terms of getting ATP, because, when people think of breaking down something for energy, they usually think of glucose, since that's what we focus on in a lot of our courses, especially the intro level bios. So now, comparing on this slide, you see an example in this table of the balance sheet, meaning the breakdown of the intermediate molecules that help with getting us more ATP, and then the ATP yield in the final column. So this is looking at the oxidation of one molecule of stearic acid, so the one molecule lipid breakdown. And you notice it's an 18-carbon lipid, and it gives you 120 ATP molecule yield from one oxidation. Now, we mentioned earlier the fact that lipids, of course, especially fatty acids, make for excellent storage of energy and breakdown of energy because they have so many carbons. Whereas when you compare it to sugar, if we look at glucose, well, how many carbons do you have in a single glucose molecule? You have only 6 carbons compared to, in this example, 18 in this particular lipid. Now, with each of those glucose molecules, when you go through oxidation, you only get approximately 32 ATP yield. Sometimes you'll see it listed as 32 to 36, sometimes even as much as 38 if you're looking at a prokaryote, but ultimately we usually estimate it around 32 ATP from a single glucose. So now, even if you then want to, make this truly comparable to the lipid example, and you say, well, the lipid is 18 carbon, so let's make the glucose 18 carbons as well. Well, you would need 3 glucose molecules to make the 18 carbon, and so that would yield 3 times 32, which would still give you a lot less ATP than the lipid, because that would give you 96 ATP from breaking down or oxidizing glucose as 18 carbons, whereas the lipid you're getting 120 in that particular example. And, if you think about it, part of the reason for that difference is that the fatty acid is all hydrocarbon, except for the carboxyl group at the end, that polar little head. Whereas when you compare to the sugar is already partly oxidized because it has oxygen-containing groups within it. And so since the oxidation of fuel leads to the reduced electron carriers used in the electron transport train, a more reduced fuel, such as the fatty acid, that can be oxidized further than the partially oxidized fuel, such as the carbohydrate. Okay, now all of that part, you don't have to memorize any of that or anything. That was just for your clarification. But I want you to really, what you should grasp from this slide is how lipid oxidation will give you a lot more ATP yield than glucose or sugar oxidation. Okay? And the other thing that I want you to gain from this is the other interesting benefit of oxidation of lipids. Not only does it give you more ATP, then oxidizing the sugars. But water is produced in the oxidation of fatty acids. And that's very interesting in terms of the production of metabolic water, because this can be very useful for organisms that live in desert environments. Okay, and so you're going to get more water out of this oxidation of fatty acids than you would get out of the complete oxidation of carbohydrates. And that's important for organisms like camels and for kangaroo rats that live in very arid areas, and they need to produce, an adequate amount of metabolic water as their main source of water for their everyday life. And what you'll notice in animals like that, their response to the arid conditions is not only to make more metabolic water with lipid oxidation, but also to have a reduced output of urine. Okay, so very, very interesting little side aspect of oxidation of lipids. Now, when we talk about the oxidation of fatty acids, I do want to point out that unsaturated versus saturated fats or fatty acids can make a difference in terms of the ATP yield. So unsaturated fats do not generate as much ATP compared to their saturated fat counterparts. Basically, this is because when you have the cis-trans isomerization or the double bond present in the unsaturated fats, this tends to be unfavorable targets in terms of some of the enzymes that would be used in oxidation. So some of the enzymes will not be able to bind or to work on this kind of a molecular arrangement. So you end up having some of the steps in terms of oxidation being skipped. So usually it's the acyl-CoA dehydrogenase step that ends up being skipped. And when you skip one of the beta oxidation steps, you then have less FADH2 produced, or NADH. And when you have the less FADH2, you then end up having less ATP generated. But the key point here is when you're looking at unsaturated versus saturated fat oxidation, unsaturated fat oxidation will generate much less ATP. Now that you're all experts in terms of the oxidation of lipids, I do want to mention one other thing about the oxidation of lipids, and that is the byproduct called ketone bodies. And those of you who have ever heard the fad diet of keto diet, that is related to this concept. So what ketone bodies are, they're basically water-soluble products that are related to acetone, and they're produced when there's an excess of acetyl-CoA from beta oxidation. And beta oxidation, as you know, is the metabolism of lipids and fats. Now, this is basically occurring when there's not enough oxaloacetate available to react with the large amounts of acetyl-CoA that could be entering the citric acid cycle. And so the oxaloacetate then in turn arises from glycolysis because it's formed from pyruvate through pyruvate carboxylase. Now those steps, you don't have to memorize that, only, the key points are on the slide already written down for you. But that was just to clarify, some information. That's basically how you'll encounter it in your textbook. Now, I want to point out that these ketone bodies, they're produced by the liver mitochondria, and then they can end up transported to other organs, because as we mentioned in their definition, they are water soluble, which means moving them around, transporting them is very, very easy within the body. And then they can be used for energy, basically, having that acetyl-CoA converted having the ketone body converted to acetyl-CoA and then have it enter the citric acid cycle to produce ATP for you. Now, what can cause them? There are a few different things. like I mentioned before, if you think of the keto diet, then that can help you remember that one of the causes of ketone bodies is if you have a high intake of lipids, high intake of fats, and a low intake of carbs. Also, if the body goes into starvation mode or if someone has diabetes, both of those can also cause ketone bodies to be formed. So basically, if you think about it, starvation mode is going to cause an organism to break down their storage of fats as their energy source. And that means breaking down fats is beta-oxidation, which will then lead to ketone bodies. And with diabetes, interestingly enough, it's not the lack of carbohydrate intake, but it's basically the inability to metabolize them. So it's kind of the equivalent of having low carb intake, and so the body then wants to use lipids or fats instead in terms of their energy source. So basically, anytime you have a lot of lipid oxidation going on, you'll accumulate ketone bodies. So this is another one of those slides where you can just listen to the lovely sound of my voice first, and then there will be a recap slide of the actual notes that I want you to write down from what I said coming up next. Now, when we talk about lipids and lipid metabolism, to everything in science and in life, there are two sides to the story. So far we focused on the breakdown or the oxidation of the lipids, but on the other side of that story is the biosynthesis of those fatty acids and lipids. Now, this will be different, of course, since it's the opposite effect or opposite event in a way. The first difference that I want you to note is where these events are taking place. So when we talked about beta-oxidation, you know that was mainly occurring in the mitochondria, whereas now for fatty acid synthesis, instead it will take place in the cytosol. Okay, so now we're looking at an anabolic reaction in the cytosol as opposed to a catabolic reaction in the mitochondria. Now, when you have this biosynthesis, basically what's going to happen first is that the acetyl-CoA has to get transported to the cytosol, and then once it's in the cytosol, that acetyl-CoA gets carboxylated, okay? And when you have that carboxylation, what's produced is known as malonyl-CoA now, instead of acetyl-CoA. And that malonyl-CoA is a big-time intermediate that will serve as the starting point in biosynthesis. So that's that second bullet point that I have here. And it's the starting point because then what happens is you'll get the growing fatty acid chain with each cycle getting the carbon atoms of the malonyl group getting added on top of each other. So basically, 2 of the 3 carbon atoms of the malonyl group of that malonyl-CoA end up added to the growing fatty acid chain, and you have this happen cycle after cycle, accumulating into a long-chain fatty acid. And the way that this is occurring is thanks to an enzyme that is appropriately named fatty acid synthase, because basically when you think of any enzyme names, they're usually pretty straightforward with what they do. So this is biosynthesis of fatty acids, so it is fatty acid synthase. Now, what happens, interestingly enough, is you'll notice in this figure that it is palmitate that is produced in biosynthesis of fatty acids. And that's a 16-carbon long-chain fatty acid. But as you know, there are a lot of different fatty acids. There's not just palmitate, and there's ones that are longer than that. And so what happens is each time it's palmitate that is first produced, and then that gets modified in chain lengthening modifications that will happen in the ER and the mitochondria. Okay, so make sure you circle star highlight the name palmitate as the product, the fatty acid product of biosynthesis that then gets modified to make the other fatty acids that we've been encountering throughout this lesson. Now, as promised, this is the recap slide of the main notes of what I was just saying that gives the answers to the previous slide. So an interesting point to make when it comes to the biosynthesis of fatty acids is that some fatty acids actually can't be synthesized by our body, because when it comes to synthesizing fatty acids, and certain fatty acids need the insertion of double bonds, mammals can't introduce a double bond beyond carbon atom number 9, counting from the carboxyl end, because we lack the enzyme necessary to introduce those particular double bonds. So you end up having what's called essential fatty acids, which are fatty acids that our body cannot synthesize because we can't insert those double bonds where they need to be. So we have to obtain them from dietary sources. And the two examples that I want you to know are the omega-3 and omega-6 fatty acids, which you should know as linoleic acid and alpha-linoleic acid. Okay, so remember that linoleic acid is omega-6, meaning that the double bond is found over here at the carbon 6, and the alpha-linoleic acid is omega-3 because you see the double bond on carbon 3 instead. Now, it's important to make sure you have plenty of those essential fatty acids in your diet because, like we just mentioned, you cannot synthesize them in your own body, which is a problem that you can't synthesize them. It's a problem that you can't synthesize them because there are precursors of other lipids, including prostaglandins. And so you'll see throughout this lesson why it's so important that you have these precursors of linoleic acid and alpha-linoleic acid. Now, before we move on to the biosynthesis of some other lipids, I just wanted to give you this comparison table found in your textbook that compares fatty acid degradation and biosynthesis. It mainly focuses on some differences, but as you can see, there is a lot of terminology that's similar between them, even though they're opposite types of reactions. So the first difference you notice is that in degradation, the product is acetyl-CoA, whereas biosynthesis relies on acetyl-CoA as the precursor. You'll also notice that whenever you hear malonyl-CoA, sorry, I keep calling it Ca, malonyl-CoA, please think of biosynthesis because malonyl-CoA is the starting point of fatty acid biosynthesis. It is not involved in the degradation or oxidation process. And then going through this listing, you see some of the differences in terms of NAD, FAD, NADPH, and various other steps. But the main thing that I want you to focus on is circle star highlight malonyl-CoA in terms of being the starting point for biosynthesis and not being involved in degradation. Also put little stars next to how degradation you have acetyl-CoA as a product, whereas in biosynthesis it is a precursor. The other aspect that I want you to circle star highlight is the location. So remember we mentioned degradation or oxidation will occur in the mitochondria, whereas biosynthesis will be occurring in the cytosol. The next kind of biosynthesis that I want to talk about is the synthesis of acylglycerols and compound lipids. These are lipids that are derived from fatty acids and metabolites of fatty acids, such as acetoacetyl-CoA. Basically, with the biosynthesis of these, you'll see it take place in the ER of liver cells or in fat cells, which are called adipocytes. And the term that I want you to associate in terms of synthesis, basically synthesis products when we're thinking of compound lipids, specifically the sphingolipid, synthesis is the term ceramides. And a lot of times you may hear this term in association with cardiovascular diseases. Basically, ceramides are products of sphingolipid synthesis that are bioactive sphingolipids that are involved in being part of the structure of plasma membranes and are involved in cell signaling between cells. And of the many different types of lipid metabolites that can kind of collect sphingolipids that are, specifically ceramides, are basically very damaging to blood vessels, and to the heart. And so when you see ceramides that have been accumulating in tissues, specifically blood vessels or the heart, you basically know that your patient is having some sort of issue with cardiovascular disease, and it can be a sign of things like hypertension, heart failure, atherosclerosis, which we're going to show again in a few slides when we talk about cholesterol. And so basically, serum levels of the ceramides are used clinically as like an indicator of really bad heart problems. Like I just said, things that show some sort of damage to your cardiovascular system, things like hypertension, heart failure, and the clogging of arteries. So please make sure you associate the term ceramides with two things. You should associate it with sphingolipid synthesis, and you should associate it with serum levels being used to indicate cardiovascular problems, cardiovascular diseases. So the last lipid biosynthesis that I want to briefly mention is the biosynthesis of cholesterol. Now, the first thing that I want to talk about in terms of this biosynthesis is the ultimate precursor of all carbon atoms in cholesterol and other steroids derived from cholesterol. And in the next slide, we're going to go over some of those other steroids. But for now, the key here is that the ultimate precursor of all of these molecules is the acetyl group of acetyl-CoA. So you see acetyl-CoA being the start of this biosynthesis pathway. The main steps are then mapped out here in this figure and kind of summarized over here so that you can see each of the terms associated with cholesterol biosynthesis. As you see, you start off with acetyl-CoA, and 3 acetyl groups ultimately produce mevalonate. And I constantly mispronounce that word, so I apologize in advance because I will stumble a few times sometimes when I say it. And then you have the decarboxylation of that mevalonate to produce this 5-carbon isoprene. And isoprene units are involved in the biosynthesis of ubiquinone. So if you've ever seen supplements called coenzyme Q sold in stores, which is associated with kind of helping your mental acuity, kind of helping you think and get through the day, that's related to isoprene units. And then you see the isoprene units then condensing to form squalene. And from squalene, you then get the conversion into the 27-carbon atom cholesterol. Okay, so you see that you've gone down to the C27 over here with cholesterol. And the enzymes involved in this process, if you ever see enzymes called HMG-CoA reductase or HMG-CoA synthase, those are involved in the conversion of the acetyl-CoA all the way through to making cholesterol. Now, as I mentioned in the previous slide, cholesterol is a precursor for a lot of different steroids, and these are very important hormones in your body. And the 5 main groups, the 5 major classes of steroid hormones that cholesterol is the precursor of, are the progestogens, the glucocorticoids, the mineralocorticoids, androgens, and estrogens. So this includes things like the sex hormones, testosterone, estrogen, progesterone, and it also includes things that are mineralocorticoids, or glucocorticoids, which are hormones such as aldosterone being a mineralocorticoid and cortisone being a glucocorticoid. And I want you to note any of these steroids and any of these hormones that cholesterol is a precursor to are pretty nasty things in terms of if you ever have to take supplements or any kind of medication with these. So please do be careful, because sometimes you have to in terms of treatment, or you think you have to in terms of treatment. But I do want to warn you, any of the ones listed on here can have significant impacts on your body and especially on your mental health. So be very careful of anything that modifies these particular hormones, including things like birth control or T boosters. If you're into gym working out, be very, very careful messing around with any of that stuff, because it can do significant damage to your body, and again, especially your mental health and moods. And since we have been mentioning with a lot of the biosynthesis and metabolism where exactly these things will occur, I do want to point out that the smooth ER, okay, make sure to write down the smooth ER, is the main site for both the synthesis of cholesterol and the conversion of cholesterol to the other steroids that you see on this slide. So please make sure to write the smooth ER. Now, even though I think, of course, all of my slides are very important slides, this one is especially important for your daily life and health. So I'm basically going to give you a transcript of what I'm going to say on this slide as a recap on the next slide. So again, don't worry about trying to jot it down. You'll have the notes written for you on the next slide. Now, one of the last things I want to talk about today is lipid transport. And this involves lipoproteins, which are terms that you've probably heard before. The different type of lipoproteins that you encounter, basically as being package transport for cholesterol in the bloodstream, are known as VLDLs, IDLs. So VLDLs for very low density lipoproteins, IDLs, which are intermediate density lipoproteins, LDLs, which are low density lipoproteins, and HDLs, which are high density lipoproteins. And what I want you to do, we're going to focus on HDL and LDL, because these are the two that play major roles in terms of your health, specifically your heart health. So what I want you to do is I want you to circle the H of HDL and write healthy. So when you hear HDL, think of healthy DL, and that's what you want a lot more of in your body. And for LDL, I want you to circle that first L and write the word lousy, because the LDL is bad. It's lousy. You want lower LDL. Okay, so you could also do that for the H and I. For HDL, the H can stand for high, because you want a high HDL and you want a low LDL. The reason that is because HDL regulates lipid storage and excretion of cholesterol, meaning, as you can see here, HDL carries excess cholesterol back to the liver to be broken down and removed from your body. Whereas LDL is the opposite. LDL will carry cholesterol to your cells, and it basically causes a collection of plaque in your blood vessels and reduces blood flow, so that you end up getting things like heart attack or stroke. Okay? And so it's very important to have high HDL to get rid of that cholesterol from your body, and low LDL because you do not want the accumulation of cholesterol forming plaques within your blood vessels. That's very bad, very dangerous. Okay? And when you get the collection of that plaque, it's called atherosclerosis, which is a major form of heart disease. Now, in order to help you get high HDLs and low LDLs and have a lot better health in your life, some of the dietary things that you can do is have a lot of fruits, nuts, whole grains, and fish like salmon. And also drinking green tea helps regulate your cholesterol as well. And as you can see in this figure, some of the things that hurt your cholesterol and will give you low HDL when we want high HDL are things like smoking, sitting around, not exercising, eating a lot of junk food. Also, if you have excess weight, or if you have uncontrolled diabetes, or even some genetic issues, that can all contribute to problems in terms of low HDL, which is very problematic because, again, the goal is to have high HDL, which helps you remove the cholesterol, break it down, get it out of your body, whereas LDL will do the opposite. Like I said, here's a recap for you to pause and write down all of the notes that you need from the previous slide. So the last thing that I want to talk about today is the hormonal control of your appetite. Some of the hormones that play a key role in this will be ghrelin, cholecystokinin, insulin, and leptin. And I will have a recap slide on the next slide so that you can see the spelling of everything and so that you don't have to worry about trying to jot down notes. Now, some of the ones that I just mentioned are on this figure right here, so you can see ghrelin, leptin, and insulin. The ones that we consider short-term effects are ghrelin and cholecystokinin, which I don't believe is on this particular figure. And then the long-term effects are controlled by leptin and insulin. Now, the stimulation or repression of your appetite is mainly controlled by neurons of the arcuate nucleus in the hypothalamus, part of your hypothalamus. And when we look at these, the first ones that I want to focus on are ghrelin and the cholecystokinin that I had mentioned. And the reason why I do this in pairs is that they have opposing functions, kind of like you'll see in a minute with the other pair that we'll talk about. So when I say opposing functions, it's because ghrelin is going to be produced by your stomach when your stomach is empty. And if your stomach is empty, then the ghrelin is going to say, hey, I need to signal that we're hungry, so you need to eat something. Whereas cholecystokinin is going to be the signal for being full. Okay, so they have opposing functions. So once you've eaten, after you've gotten the signal to eat, you're now going to be full or feel full thanks to the cholecystokinin. The other pairing that I want to talk about is then the long-term control, which is insulin and leptin. And so insulin is involved in regulating glucose uptake in many tissues, including your fat tissues. And when we talk about insulin, it will be able to stimulate the production of leptin. So they're working together. Okay. And when you have higher leptin levels, if you have a signal of higher leptin levels that reaches your central nervous system, this will tell you to decrease your appetite. Okay, and so when you see studies on obese mice, they're what you call, there are loss of function mutation, leptin resistant mice, basically mice that do not have the ability to have leptin receptors and have leptin detected or properly synthesized in their body, which means that they can never get that signal to decrease their appetite, which is why they end up obese. Okay, so leptin is involved in decreasing appetite. Now, that's why leptin you can write as an appetite suppressant. That might help you remember a little easier what its function is doing. It's basically that it binds to receptors and depresses or gets rid of some of the neurological signals that would stimulate appetite. And what they found is if body fat decreases because you have a decrease in appetite, then the levels of insulin and leptin will also decrease. And that gives you a long-term effect of a reduced appetite and lesser fat within your system. Okay, and like I said, we're going to have a recap slide now because that was a lot of information. So as promised, here is your recap slide so you can pause and write down these important notes. And that is it for today. As always, if you have any questions, any concerns, please contact me in the Remind app. Thank you and have a great day.